All right. Hi, everyone. Thanks for joining. I'm going to give the YouTube stream just about 30 seconds or so, just to make sure stragglers are coming in, and then we're going to get started. All right. Excited to be doing a live demo for all of you today. All right, let's just get started then. Hi, thank you so much for joining me this afternoon. Today, I'm really excited to be sharing BioBits and doing a live lab with you today. My name is Dr. Huang, and BioBits and this lab that I'll be doing today is actually something that I developed in my PhD when I was at MIT. And last year, when I graduated from MIT, I actually joined up with Mini-PCR Bio. So for those of you who don't know who we are, Mini-PCR Bio is a small company based out in Boston. We make biotech equipment as well as really innovative curriculum for you to be able to use this in your classrooms. And today I'll be showing you an example of one of our really innovative curriculum to cover gene expression that really hasn't been done in this way before. So, before we get started, just a couple of housekeeping notes. The live chat is going, so if you have any questions, feel free to direct your questions there, and our moderators will answer them for you. And there's also quite a few resources linked down below in our YouTube description. All right, let's take it away. So, we're going to take it a step back. Most of us know what DNA is, and that we each have a unique DNA sequence that makes up who we are. But how does DNA do this exactly? DNA is actually the blueprint for life. It's the instructions needed for us and all living things to survive. Now, these instructions from DNA are used to build proteins. Proteins are the tools that provide structure and function for all of the cells in your body to keep you alive and make you who you are. But what is reading these instructions and what is building these proteins? So, there's a middle step with a molecule called RNA, and it's like the messenger. It carries information from DNA to protein. And since this flow of genetic information was so fundamental and so important to how life functions in biology, that it is known as the central dogma of molecular biology to indicate its importance. So whenever you hear someone refer to, the central dogma, this is what they're talking about. So, when you're first teaching or learning about DNA, this abstract diagram that I'm showing here is probably a little too simple. But when you open up your textbooks, you probably see something like this, which can be pretty confusing and overwhelming for someone learning about it for the first time. Manipulatives and 3D models can be really helpful to students to visualize these molecular concepts and make them a little less abstract. So today, I'm going to show you another way that we can visualize these molecular concepts using a hands-on lab activity that lets students actually see real DNA and real proteins in action. So you might be wondering, how are we actually going to be able to see all these things at the molecular level? So for today, we'll be using something called fluorescence. So if you have ever drawn anything with a highlighter and then shown a black light on it and it lights up, that's fluorescent. Something that is fluorescent can absorb UV or other types and wavelengths of light and then emit that energy back out as visible light that we can see. So these highlighters contain a fluorescent chemical, but certain other proteins can also be fluorescent. Scientists often use fluorescence in biology so they can track different parts or processes in experiments, like shown here. They are lighting up cell structure like the mitochondria or the nucleus. So for today, we'll be using both fluorescent chemicals and proteins in this lab to light up the central dogma so we can actually see when RNA and protein get made. And as an aside, teachers, if you are curious about, how this fluorescence in proteins actually work, we're actually currently working on a new BioBits lab that's going to cover this topic, so stay tuned. Alright, so throughout this demo, I will be briefly reviewing this process for the audience members out there who might not have learned this in class yet. So for the central dogma, you can think about this like cooking. So think about DNA is your cookbook, and each protein is a different dish that you're making. Where does your recipe come into all of this? Each set of instructions is called a gene, which is found in the DNA. And all of us each have over 20,000 genes. It's like we each have a cookbook of 20,000 recipes. And each gene makes a different protein that we need. The process of how the protein is made from a specific gene is known as gene expression. And this is made up of two main processes. The process of DNA going to RNA is called transcription, and the process of RNA going to protein is called translation. And again, as I do the lab, we'll be reviewing this in more detail later. But first, I wanted to point out that transcription and translation happens typically inside cells. So it's happening in all of your cells right now. For eukaryotic cells, transcription, which is DNA going to RNA, happens in the nucleus of the cell where DNA is stored, as shown here. And then after you get RNA, it leaves the nucleus and goes into the cytoplasm where the process of translation, which is RNA to protein, happens. So, scientists and researchers have been using cells as workhorses to make proteins for many decades, but I'm currently sitting in my bedroom. I'm not at lab. Obviously, I can't really grow cells in my house and use them for gene expression. So, what do we need? What can we do? So that's where BioBits comes in, and this idea of cell-free protein synthesis. So I actually did this a lot in my PhD, and I actually did a webinar last Monday covering, what cell-free technology is and how it's used. If you want to see a little bit more about that, you can check out the link down below in the description. But for today, I'm just going to review this really, really briefly. So back when scientists were still figuring out how gene expression worked in the 50s and the 60s, they found that you could take cells, in this case a bacteria cell, bust the cells open, and then scoop out the molecular machinery. So things like we're going to talk about later, like RNA polymerases, ribosomes, transfer RNAs. And then you can actually use these parts outside of its cell. And if you add DNA to it, it still can make proteins. Kind of the analogy that I described before is think about a car, and you actually pop the hood and take the engine out of the car. Even if you took the engine out of that car, you could still hook the engine up to other machines and get it to run. It doesn't have to be in the car in order to work. Same thing here. And then to make it even easier for students to handle, we freeze-dried these reactions into solid pellets, kind of like when people freeze dry ice cream into a solid chunk so it doesn't melt at room temperature. Same thing. So this is what BioBits is: freeze-dried cell-free reactions specifically for educational use. They're super easy to use because there's nothing you need to keep alive. You don't have to grow cells. You don't have to transform your DNA and hope it gets into the cells. All you have to do is just add water directly to the pellet that has DNA in it, and I can even just make proteins right now in my bedroom, which is what I'm going to do. So let's switch over and just showing you here. Not sure if this shows up on camera, but on the bottom of these tubes right here are the freeze-dried cell-free pellets that I was just talking about. So, these are the things that have all the molecular machinery that you need for transcription and translation. So, I'm going to add the DNA with the genes that we want to express, and then we'll get the proteins. So I'm going to add the reagents now, and then while we wait for the reactions to incubate, I'll explain what is happening. Quick disclaimer, everything I am using today is perfectly safe to use without gloves or goggles, which is why I'm not wearing them right now. However, just make sure you follow your local guidelines if you do this lab in your classroom or at home. Alright, so we have 4 tubes here that we're going to add reagents to. Okay, so in this first tube, we are just going to add water. Now, this water does not have any DNA in it. So, as I'm doing this, you can think about, why are we just adding water to a tube if there's no DNA in it? There we go. Alright, to tube number 2, we're going to be adding DNA. And really quick, I'm just going to bring up a little chart here so you can follow along to see what I'm doing. So in DNA, or in tube 2, I'm adding a DNA called DNA-A. And again, I will explain exactly what that is in a bit after I finish up here. And then in tube 3, I'm adding that same DNA, DNA-A, but I also will be adding an antibiotic called kanamycin. And what kanamycin does is that it binds to the ribosomes and interferes with them. So, I'm not going to spoil the surprise yet, but you can start thinking about what happens when the ribosomes don't work. Alright? And in the last tube, I'll be adding DNA again, except this is a different type of DNA called DNA-B. And we're not going to know what DNA-B is, because tube 4 is actually going to be our inquiry tube. So we're going to look at the results from tubes 1, 2, and 3, and then use that to figure out what happened in tube 4 and what DNA-B actually is. Alright, so now that I've added all my DNA and my reagents to my tubes, you can see that the pellets are now rehydrated. There's liquid at the bottom here, and let's just take a peek at what they look like at time 0. I'm going to explain what this is in a bit, but this is just what we're going to be using to visualize the fluorescence. So if I stick it in here, turn off my light, show this to the camera, you can see the tubes, but they're not lit up. Nothing is glowing, which is what we expect at time 0. Alright, so to incubate these tubes, we're going to stick them at 37 degrees Celsius. So what I'm going to be using today is a mini PCR thermocycler set at 37 degrees Celsius, but you can use any incubator capable of that temperature. In fact, if you don't have an incubator, body heat works just as well. I've done tons of these reactions and just stuck them in my pockets or under my arm or in my fist to warm them up. So you don't need a lot of equipment to do this lab. All you really need is a pipette and something to visualize it in. And that's it. Alright, so we're going to let the tubes incubate there, and while the tubes are incubating, as promised, I'll take you through to what's going on. Alright, if we go back to our slides. So, going back to our analogy, when you want to make a recipe, first you need to look it up in your cookbook and copy it down on a piece of paper. When you do that, you are transcribing the recipe, a process called transcription. Something, however, needs to be doing the actual transcribing for the gene, and in this case, it's a molecular machine called the RNA polymerase, shown as this green blob in this diagram here. Now, the RNA polymerase takes the DNA, shown in here in blue, and begins copying the gene letter by letter, nucleotide by nucleotide, building up this molecule here called the messenger RNA. So that's the copy of our gene, a copy of the instructions that we need to make the protein. When you're copying from your cookbook, though, how do you know when a recipe starts and stops? You don't want to accidentally copy another part of a different recipe. So when you're looking at a recipe in a cookbook, usually you start with the title and you go until the recipe says something like enjoy or bon appetit. And so the same thing is true in a gene. The gene has a section called the promoter. It indicates the start of the gene. It also has a section called a terminator, which indicates the end of the gene. The RNA polymerase binds to the promoter, and starts transcribing there and goes until it reaches the terminator and stops. The result is that you have the messenger RNA that contains this middle section here that contains your recipe information, how to make your protein. Now, RNA is not normally fluorescent, so how are we supposed to see it? For this lab, I engineered a special trick into the gene so we can actually make it so we can fluorescently label it. So that trick is the sequence here called the aptamer. And how this works is when transcription happens and you get RNA, that particular section of RNA will fold up on itself and form this little pocket. This pocket will bind a chemical that we've also freeze-dried into the BioBits, and when this binding interaction happens, green fluorescence is emitted. So when you see green, you know that you must have RNA, which means that transcription must have been successful. Again, keep in mind, this normally does not happen in your cells. Your RNA isn't glowing. The rest of the RNA, in fact, is not folding up and glowing. It's just a special trick we engineered in this lab in order for you and your students to be able to see RNA. Alright, so let's switch back to our demo here and see how our tubes are doing. Alright, let me take a look. Alright, so they're glowing now. Give me a second. My mini PCR cord just knocked out. Alright, so let's take a look here. So how do we actually see fluorescence? Like I mentioned before, in order to see fluorescence, you need to excite the fluorescent molecules or proteins with the right wavelength of light. So in this case, blue LED light. So I will be using this box here. You can put your tubes in right up here at top. It's made out of cardboard, and it has blue LEDs that can shine up at your tubes, and then there's a orange filter in front that you can use to filter out blue light so you can actually see what's going on in your tubes. So we call this little box P51. So P51 stands for Photograph 51. This is actually our tribute to Rosalind Franklin. So Photograph 51 was Rosalind Franklin's photograph X-ray crystallography that really helped contribute to determining DNA structure and the fact that it was a double helix. Since this was such a huge visual contribution to the history of molecular biology, for our visual molecular biology labs, we decided to name this after her. In fact, if you open this up, we actually have a little picture of Rosalind Franklin in here. And this P51 box can also actually be used for a variety of other labs that we have. So we have other labs that cover things like DNA structure or enzyme activity, and these are relatively inexpensive. They're $28 each, or I think $170 for a classroom set of 8. But any other blue light illuminator will actually do. If you have one of our, where is it? If you have one of our blue gel electrophoresis boxes, these actually have a blue light illuminator built into them as well. So if you already have these to run gels in your classrooms, you could actually use this to visualize your BioBit reactions. Alright? Alright, so let's take a look at our tubes now. Put them in here, the box, turn on the light. Alright, so I'm going to turn the light off here so we can actually see it. And if I hold it up to the camera, yeah, there you go. So you can see that tube number 1 is blank, and that tubes number 2, 3, and 4 are green. So in those tubes, since we see green, it must mean we have RNA and that transcription was successful. And as a difference between these 3 tubes, we'll learn about that more as we visualize the second process, which is translation. Alright, so I'm going to turn my light back on. Switch back to slides. So, for translation. So now that you have your copied or your transcribed recipe, you're going to take it down to your kitchen where you'll use the copied recipe to cook your dish. You need to read the recipe and then translate that to cooking actions. Just like we had RNA polymerase to carry out transcription, we need another molecular machine to be our chef and actually make the recipe. For translation, this is the ribosome. The ribosome takes the messenger RNA, or mRNA, and reads the nucleotides, and then translates that into the language of proteins, which are amino acids. These things in here, the blue squiggly things, they're called transfer RNA, and they're like the Google Translate for the ribosome. And I'm not going into detail here, but teachers, this is a great point to talk to your students more about the genetic code and how codons work exactly, but we're not going to get into that for today. So the ribosomes and the transfer RNA will build the protein one amino acid at a time, stringing them up like beads on a string until the protein is done. So, like the RNA polymerase, the ribosome also needs to know where to start and where to stop. Where's step 1 and where's step 10 or whatever the final step is. So the ribosome also needs its own set of start and stop indicators called the start codon and the stop codon. This info is coded in the gene and gets transcribed over into mRNA, so the ribosome can read it. The ribosome is going to find the start codon on the mRNA and begin translating and adding amino acids to the chain until it gets to the stop codon. And everything in between the two is the protein coding sequence. And that's the actual recipe that the ribosome is working off of to build the protein. All right. So, how do we actually visualize proteins? Most proteins aren't visible by eye. But again, for this lab, we actually want you to see it. So there are proteins out there in nature that are naturally fluorescent, like ones that you'll find in jellyfish or coral, as shown here. So for today's lab, we'll be using a gene found in coral that expresses a red fluorescent protein. So when this particular protein coding sequence is transcribed to RNA and then translated to protein, it'll be fluorescently red. Alright, so let's take a look at tubes now. So the way this lab is set up is after you see transcription on the first day, you'll leave your tubes alone. They don't actually have to be at an incubator overnight. They can just be lying on the bench or on the table at room temperature. And when you come back, you'll be able to see translation. So let me get this going, turn off my light again. So I started these tubes yesterday, so these are the results that you see today. So what we see here is that tube number 1 is still blank, tube 2 is red, and tube 3 and 4 are blank. So in the case of tube 2, since we see red, it must mean we have the red fluorescent protein that I just talked about, and that translation was successful. Alright, lights back on. So let's review our results and figure out why we saw what we saw. Alright, so just to review, tube number 1, we didn't see anything. Tube number 2, we saw green, then red. Tube number 3, we just saw it go green. Tube number 4, same thing, we saw it just go green. So, what's going on? And I'm going to now walk you through it tube by tube so we can see what's going on. Alright, so in tube number 1, we just added water, right? So we didn't see anything, and in this case, no DNA was added, which means obviously if you have no DNA in the tubes, no transcription or RNA will happen, and no translation or protein will be produced. So this is not that interesting, so why do we have this tube? This is our negative control. It acts as our baseline for our experiment, so we can actually compare our other tubes to, so we know if something actually happened in those other tubes. It's a great way to show students that, why do we need negative controls in scientific experiments? And so yeah, it's just our baseline. Tube 2. So in tube 2, we saw that it went green and then red. And in this tube, as you remember, we added something that I called DNA A. So since we saw both green and red, that means both RNA and protein must be present, which means both transcription and translation happen. And for that to happen, DNA A must have contained a gene that had all the needed parts, right? It had the promoter and the terminator for the RNA polymerase to do transcription, as well as the aptamer sequence so we can see it. And then there's also the start, stop, and the protein coding sequence for translation. So all the parts of this gene was needed in order to get both RNA to see green and the protein to see red. Okay? So tube 3 is where it gets really interesting. In tube 3, we saw green, but then no red. And in this case, we added DNA A, which is the same DNA that we added to tube 2, but we also added an antibiotic called kanamycin. And if you remember what I said before, kanamycin binds to the ribosomes and causes them to mistranslate and interferes with translation. So, quickly to review, we did see green, so that means RNA must be present, which means transcription must have happened. And again, in order for that to happen, this gene must have had the promoter, the terminator, and the aptamer sequence, right? It's the same gene as, the one that we added to tube 2. But there's no red, which means there's no protein, which means that translation didn't happen. So take us a couple seconds and think about what part of the process could have been broken. I'll give you guys like 3 to 5 seconds to think about it. So looking at this, it could mean one of two things, right? Either our recipe is broken, right? Something is missing in our gene here, or the machinery is broken, right? The chef is sick. And if you remember, again, we added the same DNA as tube 2, so it can't be the DNA or the gene, right? This worked just fine in tube 2. So it's the same DNA, it's not something wrong with the gene or the recipe. But we did add kanamycin, which, like I said, interferes with the ribosomes. And if the ribosomes don't work, they can't translate. So you don't get protein, and if you don't get the red fluorescent protein, we're not going to see red. Alright, so tube 4, our inquiry tube. And this tube was very similar, behaved similarly to tube 3. We saw green, but no red. And now in this case, in the guide, we don't tell you exactly what DNA or gene was added. We just tell you it's DNA B. So you have to use your results from the previous tubes to figure out what's going on. So let's run through it again. So the DNA you added, transcription was successful because you got RNA because you were able to see green. Again, for that to happen, that means the gene must contain all the structures you need for transcription and to visualize it, which again is the promoter, terminator, and aptamer sequence. And again, we didn't see any red, which means again, translation didn't work. So again, it's one or two things that happened. Either our recipe is broken or the machinery is broken. But now if you remember, this time we didn't add kanamycin or any other reagents besides the DNA. I only added DNA B. So clearly, the only difference here was that I added a different kind of DNA or gene. So looking at this gene structure, what do you think could have been different about the DNA that was added to tube 4 so that translation wouldn't work? Again, I'll give you guys 5 seconds to think about it and see if anyone can guess it. There is more than one right answer based on the information that you have here. Alright, so let's go through it. One possibility is that you don't have a start codon. If you don't have a start codon, the ribosome doesn't know where to start and translation can't start. The stop codon could be too early, or could be up here too early, and then the ribosome will stop before the protein can get made. There could be a mutation in the protein coding sequence, like a typo, and then the ribosome will end up making the protein wrong, and then it might not work or fluoresce red. There could be a completely different protein coding sequence in here, so the ribosomes might have made a completely different protein that doesn't glow red. And so all of these are right answers based on the information you have from your lab. And if anyone is curious, what we actually did is just take this entire thing out. Start codon to stop codon and the protein coding sequence. Alright, now you've seen gene expression in action. You've seen how DNA is transcribed to RNA through transcription using RNA polymerase, and then the RNA is translated to protein using ribosomes. And then by lighting up the RNA with green and the protein with red, you could actually see each step of the process. You also got to learn different ways about how this system can be broken. As you saw in tubes 3 and 4, if any parts of the molecular machinery, like the ribosomes in tube 3, aren't working, then this process doesn't work. And similarly, in tube 4, if any part of the gene structure is missing or incorrect, it would also not work. And overall, I hope this was clear that this was a really easy lab to do. I was able to do it live from my bedroom with just, a pipette and a blue light illuminator. It's been really great because this activity was something I developed in my PhD, and I've gotten to see it grow from my PhD lab into actual classrooms. It's always great to see students' faces light up when they see the glowing green and the glowing red for the first time. And now I'm here doing the lab to dozens of people over the internet. So yeah, that's really great kind of see this lab come a long way. Now I'm going to end with a couple of resources for teachers who are curious. So this lab comes with a very comprehensive guide and curriculum. Actually, if you go down to the link in our description, there's a link to the teacher guide, which, as well as things like classroom guides, student guides. The teacher guide contains all the worksheets and data tables. We try to make all our labs very high quality and self-contained. The idea is that your students can get everything they need from just reading the background. And all of this is free on our website. You can actually go and read through the entire lab now if you wanted to. Like I said before, we try to build in lots of inquiry for the students. They're not just passively adding reagents to the tubes, they're actually trying to figure out what's going on. The particular strength in this lab is that it's very visual. Since we had a really visual lab component, we wanted the curriculum to match the highly visual nature. So we have things like concept maps where students fill in what's going on in transcription and translation. We have things where students answer questions like, okay, in this lab, let's say you were missing transfer RNAs. Would you see green? Would you see red? What happens if you're missing amino acids? Would you see green? Would you see red? And you would go through all of that and just circle that. So a lot of really nice, highly visual questions like that. And for teachers who are teaching more advanced students who want to do a little more extensions, there's an extension here included that covers the RNA aptamer. So it delves into how researchers and scientists discovered RNA aptamers and actually how they use them in their research. They actually use them to light up structures as well in cells. So this is a great extension to learn more about how it works, and students can actually do sequence analysis of the RNA aptamer to see where it would bind up on itself. And then I also want to point out DNA Dots. So DNA Dots is a free download series that we have. These are 2-page simple explanations of common genetic techniques, things like sequencing or CRISPR, and we actually have one on cell-free protein synthesis. So the technology that BioBits was based on. So check that out. Again, link is down below. And so now, a lot of teachers submitted questions ahead of time, and so I'm going to go through now and answer the most commonly asked questions. So, common question was, how long does it take to run? So, you just saw me do it. It's a very quick lab to run. it takes students about, depending how well they are at pipetting, takes them about 10 minutes to, pipette and set up the reactions and add the reagents. And then you just, incubate and make the initial observation. So that can be anywhere from 15 to 20 minutes. So that in total on the first day is about 30 minutes. And then you come back the next day and you take your final observation. So that's anywhere from 5 to 10 minutes, depending on how much discussion you want to have. And just a note, I said next day, but you can actually come back anywhere between 8 hours to 72 hours after the fact and see your next day observations. So for example, if you set this lab up on a Friday, you could still come back Monday and see your results, and nothing will be changed. Alright, so people also asked, what equipment do you need? Like I said before, all you need is a micropipette, one for each of your lab groups, and one of these blue light illuminators like our P51 or our blue gel if you have that, or if you have your own blue light illuminator. 37-degree incubator is recommended, so if you already have a mini PCR thermocycler or your own incubator, that will work. But again, like I said, using body heat works just as well. You can just stick it in your pocket and that will work. So some people ask, why does the green signal disappear by the next day? So in tubes 3 and 4, they were green the first day, but by the next day they weren't green. So the reason for this is that RNA has a relatively short half-life. So by the time you come back the next day to see your results, the RNA has already broken down. So if the RNA is broken down, it can't form the binding interaction to give off the green signal, so you don't see it. So you only see green the first day. So why does the red fluorescent protein take so long? So transcription and translation pretty much happen at the same time in these tubes. The only difference is that the red fluorescent protein, after it gets translated and it gets made, it takes some time for that protein to fold and mature. It takes some time to accumulate enough of the red fluorescent protein that we can actually see it with a naked eye, which is why you usually don't see it. You can start seeing it around like 3 to 4 hours at 37 degrees Celsius, or you can just come back the next day and it's bright red. Logistically, how many students can use one kit? All of our kits that we sell at Mini PCR include enough reagents for 8 groups. So if you put your students in up to groups of 4, that will be up to 32 students per kit. There were some questions about the shelf life of the reagents. So currently on the bag it says that you can store this at 4 degrees or in your fridge for 3 months, and that's just a really conservative estimate from our end. We suggest you buy the kit right before you actually are going to use it in your classroom, but if you want to store your kit and have it last longer, a trick is you can actually put it in the freezer at -20, and that will double the shelf life of your reagents. It will actually last up to 6 months if you put it in the freezer instead of the fridge. How can I expand on this activity for more advanced students? Like I mentioned, in the curriculum, there are a lot of extensions built in, things like reading about the RNA aptamer or how things like cell-free work. But if you want a more hands-on expansion for your students, if you have reagents left over, a lot of teachers have told me they've had their students design their own experiments. Things like, what happens if you expose the DNA reagents to UV light for a period of time and then try to use it in your BioBits? And that would be a really fun way to explore concepts like DNA damage. And there are other things you can do, adjusting the pH, adjusting the temperature that you incubate them at, adding things like salt or rubbing alcohol. So there's a lot that you can do. If you have any specific questions or you want to run one of these experiment ideas by me, feel free to shoot me an email. I've linked the email down below in the description. It's just biobits@minipcr.com. So if you have any questions, feel free to just contact me. And lastly, will there be more BioBits cell-free labs offered in the future? And like I alluded to before, the answer is yes. The Central Dogma Lab is just the first in many labs we're hoping to release using the cell-free technology, and we're hoping to release our next one very soon, so stay tuned. And if you really like the idea of using these P51, like if you're already going to buy pipettes and a P51 for your students, we have tons of other labs as well. We have one that covers DNA structure and base pairing. So I did the Central Dogma one here today. We have a lab that covers enzyme activity. We have our qPCR lab. So qPCR is quantitative PCR. This is actually quantitative PCR is the technique that scientists are currently using to test COVID patients. So this is actually a really relevant lab to learn what qPCR is and how it works exactly. We have the chlorophyll lab, which is actually a free lab because you don't need to buy any reagents from us, especially if you already have a pipette and a P51. You just go outside and pluck a couple leaves off a tree, grind them up, throw them in your P51, and you'll be able to see chlorophyll. Don't want to give too much away, but that's also always a really fun lab. And then lastly, we have an Intro to Fluorescence lab. So if you want to learn a little bit more about how fluorescence works, this is a great lab for this. And right now, I know everyone is stuck at home and trying to teach from home. We're happy to support teachers. If you're interested in using our labs, especially these P51 labs and the BioBits lab that I just did, as some sort of distributed lab platform to do teaching and do hands-on labs at home, just shoot us an email and we can figure out how best to support you in that. And yeah, the P51 labs are just a small section of the labs that we have at Mini PCR. We also have tons of labs covering things like PCR, gel electrophoresis, restriction enzyme digests, and we have labs ranging from anything from, forensics labs to figure out who done it, to labs where students can actually take their own DNA and analyze them and figure out what genotype they are. In fact, next Monday, April 20th, same time, 3 p.m. Eastern, we are going to be doing a live demo of our PTC Taster Lab, which is where you can examine how small genetic variations can change your ability to perceive the world around us, in this case, taste. So my colleague's going to be leading this lab, and he's going to be genotyping his entire family and walking you through it. So be sure to join us on that. We'll be sending out more information later about how to sign up, or just watch this YouTube channel. All right, thank you so much for joining me. I had a blast being able to do, an experiment in my bedroom. I've really missed being in lab. So I hope that was really helpful. Again, if you have any questions, feel free to reach out to us. We're happy to support you, especially during this difficult time. And again, links to tons of different resources are down below in the description, and we'll also follow up with some more later. All right, thanks so much. Have a good day. Stay healthy. Stay safe. See you now.